This procedure prepares glass cover slips with viruses for use in atomic force microscopy and super-resolution microscopy, such as fluorescent photoactivated localization microscopy. First, clean the glass substrate. Coat the glass with a thin film of partially biotinylated polyethylene glycol grafted to poly-L-lysine. Next, treat the surface with avidin to increase the binding affinity of the biotinylated film. Now, treat the surface with biotinylated antibodies specific to the virus of interest. Then, treat the antibody-prepared surface with the virus of interest.
Unlike spin coating, this technique has the advantage of distributing the virions along the surface homogeneously in order to optimize microscopy.
Place the cover slips vertically in an appropriate sized Teflon cover slip rack and submerge them in a beaker filled with filtered ethyl alcohol. Sonicate for 30 minutes. Then rinse the cover slips extensively with ultra-pure water. Next, place the cover slips in a separate clean Teflon rack and beaker filled with 1 molar sodium hydroxide. Sonicate for 30 minutes and rinse extensively with ultra-pure water. Now dry the rinsed cover slips under a nitrogen stream and transfer them to a clean, dry Teflon rack. Verify that each cover slip is clean from any visible film or particles. Immediately after drying in the nitrogen stream, horizontally place 1 cover slip into a sterile petri dish. On the center of the cover slip, pipette the appropriate amount of biotinylated polylysine grafted polyethylene glycol in PBS. Place another cleaned cover slip atop the fluid in a sandwich method. Make sure that the volume between the active faces of the two